Good afternoon, good evening, as the place you are in. Welcome to 61st IOMP School webinar. We have started the IOMP School webinar on 11th May 2020, and this is the third anniversary. We have the 61st IOMP School webinar. Thanks to John Damilicus, President of the IOMP, for giving me the opportunity. Today's topic is very important because in 127 years of radiotherapy, we have come a very, very long way because the imaging technologies has given us not only the anatomical imaging, but biology of the tumor and biology of the surrounding tissue. And therefore, radiotherapy, there is a radical shift from a physical dose to a biological dose, from the understanding of the biology of the cancer and its related organs. So today's topic is radiogenomics and radiomics-guided personalized radiation therapy, current status, challenges, and opportunities. And this webinar is being given by Dr. V. Subramani, who has a long experience of 27 years as a clinical medical physicist, and presently he is working at a renowned national premier institute, All India Institute of Medical Sciences and National Cancer Institute as chief of the medical radiation oncology medical physics staff, and also as a teaching assistant professor in radiation oncology medical physics. After doing his MS in medical physics in 1996, he joined the All India Institute of Medical Sciences, and he has a PhD in radiation oncology in 2006. His interests are IMRT, VMAT, SRS, SBRT, everything which is modern radiotherapy medical physicist should know and should have. He is a well-known figure in India and also abroad. He is currently the Secretary General of Association of Medical Physicists of India, and he has many awards, to name few: UICC Fellowship. Then he is a member of the IOMP. Earlier he was into the Science Committee, now in the ETC, editor of the Asia-Oceania Journal of Medical Physics. Without not taking more time, I will give the floor to Dr. Subramani. Before that, I will request all the participants. The number has swelled almost 563 now, and a lot of people are joining, delegates are joining now, that the question and any questions are there, please put into the QA section so that I will collect all those questions, and I will try to put to today's speaker, Dr. V. Subramani, and we'll have a discussions and satisfactory answer from him. So now, Dr. Subramani, floor is yours. Welcome to this 61st IOMP School webinar. Welcome.
Thank you very much, Arun sir. I just share my slides.
Fine, they are visible. Good.
Good morning, good afternoon, good evening to all medical physicists across the world. It's a great pleasure, indeed my honor, to give the talk in the World Forum of Medical Physicists through the IOMP, the school webinar. Today, I will be talking on the radiogenomics and radiomics-guided personalized radiation therapy, current status, challenges, and opportunities. I do not have much to disclose of my conflict of interest. So over another 40 to 45 minutes, I will be giving brief overview on particular, the terminology rather than going into the details by giving that outline like the introduction and the background and the concept of precision medicine and personalized radiation therapy, the role of radiomics and radiogenomics and Radiomics-Guided personalized radiation therapy, current status, challenges and opportunities, then future directions, and the summary and conclusion. This is how I structured today's talk. So we all are the medical physicist community. I thought, okay, let me introduce our topic through the understanding of what is the role of medical physics in the era of precision medicine, indeed the precision cancer care. So the medical physics, as everybody very much understand, that it is a branched out from physics as an applied physics. Indeed, it is a translational science because the physics discovery and innovations are translating into the medicine for diagnosis and treatment of the disease. And the medical physicists ensure the safety and quality by means of assuring the precision and accuracy for effectiveness and use of diagnostic and therapeutic radiation. So in the context of the precision cancer care, that the subspecialties of the medical physics needs to be bridged, especially that imaging and therapy physics is an essential to bridge for the precision cancer care. Because the traditional role, as we know and we perform the everyday, the dosimetry, commissioning and treatment planning, quality assurance and radiation safety. From this, because of this, the precision, the care or the personalized medicine requirements, our roles will be expanding to the standardization. Of course, this role is being played in a very, normally through the validations and quality assurance and other, stuff in that, the routine procedure, that is the standardization, optimization and adaptation. The third is that the fourth is the personalization and integration. So this is what the very important, the concept pertaining to this topic, that the personalization of that, the radiation, the characteristics to the cancer treatment is an important role of medical physics. In this scenario, the nuclear medicine imaging and the radiology imaging and radiation oncology all together needs to be combined to work together to work towards the precision cancer care. So where are we today and what was the development from the beginning of this, the physics innovations and discovery? I just put it, the one slide, probably 125 years of, the achievements cannot be, put it in the one slide. So it is a little bit of busy, but I'm sure that everyone can understand. So after the discovery of the X-ray in 1895, so the two-dimensional X-ray imaging was used as a conventional radiotherapy. Then important, the milestone in 1972, after the introduction of the CT scanner, the 3D informations of the tumor was possible to obtain. Based on the 3D imaging, we are able to conform the dose to the target volume, which is called as conformal radiotherapy. Then the third is that one of the major breakthrough in the radiation, the physics, is in an inverse plan based intensity modulations in the year 2000. And then that image guidance in 2010. Then 2010 onwards, the particle therapy. And then that, now we are in the 2020s, very important the topics are, emerging. In fact, it is not widely used, the country like ours, but however, that importance of this topic needs to be highlighted. That was the purpose of my lecture. So in 2020s, three important techniques like the MR-guided radiotherapy and PET-guided radiotherapy, and third one is called personalized radiotherapy towards the precision medicine. So having, the MR linear accelerator, we are able to get that, the imaging, the quantitative imaging or quantitative imaging biomarkers based on which we are able to characterize biologically the tumor and then to design the treatment plan. That's called the online adaptive radiotherapy. Similarly, the PET-LINAC is an emerging technology now, and it is going to give a lots of potential for exploring that biology, based on which we will be able to design the treatment. That's called the biology-guided radiotherapy. Coming to this topic, and towards the precision medicine is one of the feature medicine, and towards that, the prediction, prevention, and personalization and participation. These are the 4 P's of medicine, and the whole, the medical community is, working towards that, how to establish the precision medicine with respect to the, the discipline. So similarly, the radiation oncology, that, the genomic, the data is being explored to personalize the treatment, that is called the personalized radiation therapy. This is what we are going to see in this, over another 40 minutes. So I'm just briefly introduce the terminology, because everybody knows that what is the precision medicine. However, there is the small definitions by that NIH NCI. It states that the precision medicine is a form of medicine that uses information about the person's genes, proteins, and environment to prevent, diagnose, and treat the disease. So similarly, the European Society of Radiology also has defined the term for what is precision medicine, like, right treatment to the right patients at the right time. That's the definition of the precision medicine. So this similar concept is being, adapted to the radiation therapy. Like, when the cancer radiation treatment move from one size fits all approach to the individualized treatment approach, which is tailored based on the individual patient's genomic profile, in addition to the image-based profile, is known as personalized radiation therapy. See, this picture shows that how the difference between the conventional medicine and precision medicine. In conventional medicine, the treatment is designed for the group of patients based on the anatomical sites, TNM stage, and histological and clinical feature. And the result of the treatment will be combined like a responder, non-responder. In case of the precision medicine, the individual treatment, individual patients level at the genomic level and omic level parameters. In addition to that, all that, the anatomical and the histological and lifestyle and behavior and medical records and environmental parameters, all these parameters will be incorporated based on a particular patient. So that treatment will be customized for a particular patient is called, the precision medicine. So this is the basic difference. So what is the need for the personalized radiation therapy? So the current practice of radiation is, like, as I told you, one size fits all approach, wherein we are the oncologists are prescribing that uniform dose prescription, which results in that adverse effect, also result in suboptimal clinical outcome. The reason is that conventionally, that the routine practice, the 3 important parameters are important for any successful treatment. One is that, the physical factors, another one is the patient specific factors, third one is that, the biological factor. In routine conventional radiotherapy, we only obtain, the important patient specific clinical factors and physical factors, and third, biological factors are not much incorporated in the conventional way. So because of this, there is an evidence that RT failure is based on that, the biological characteristics or, the process like a hypoxia, radiosensitivity, and tumor cell proliferation and tumor heterogeneity. So due to the advancement in the genomics, molecular imaging, and information technology and computer, including that imaging technology, now it is possible to obtain that, the information of particular patients of the molecular, functional, metabolic, and genomic informations now. So having all these informations, one can design that, the treatment. So the success of the treatment depends on the ability of the ability to choose the right treatment regime, that is the precision, in the right patients, that is the personalization. So it is a departure from one size fit to all to one size fit one approach. That's what it is a need for the personalized radiation therapy in current scenario. Because 80% of the patients, the complication is based on the patient specific, that is where that patient specific parameters like a genomic is going to give that, more information. So what is the radiogenomics? Because the topic is such that even, the most of the person, including me, are unable to, know earlier. I thought, okay, just to briefly introduce how that, the terminology actually meant for. So the radiogenomics is basically meant for in two contexts. One is in radiotherapy, another one is imaging. In radiotherapy, this means that a study of genetic variation associated with the response to the radiotherapy. Whereas in the imaging, it is a correlation between the imaging and genomics, indeed the tumor biology. So the radiogenomics is actually in a combination of the radiomics and genomics. And the radiomics is an imaging biomarker. Genomics is a genetic biomarker or molecular biomarker. So combination of these two will be called as a genomic biomarker. So you can see that the radiomics, the features can be obtained from that, the CT, MR, and PET CT imaging the modalities. Then the genomic parameters can be derived from the tissue sample, obtained and analyzed through the genomic test, like a DNA profiling, RNA profiling, and etc. Then the clinical data will be given. All three data will be combined. Then you can form that, the radiogenomic data. This radiogenomic data is actually plays a major role, not only, the improving for, one particular aspects of that, the cancer imaging and therapy. It is also almost from the screening to that, the treatment monitoring. This radiogenomics is being actually potentially used, such as cancer screening, early diagnosis and better staging, the prognosis of the disease, prediction of the treatment response and outcome, patient risk stratification and assessment, and indeed the personalized radiation therapy. So this is what actually the radiogenomic is. So I am not going to give much detail into that, the radiomics and radiogenomics. Indeed, I will be briefly introducing that, the basic concepts or basic terminology of, the cancer genomics or tumor biology. So you all are aware that cancer is a genetic or genomic disease. The genomic is the study of the complete genetic information of an organism, whereas a genetic is basically a study of single gene. So genes are a vital part of a DNA. So the cancer occurs when the changes in person's genome and their DNA resulting in cell growing and dividing uncontrollably. Every human body is made up of almost 50 trillion cells, 22,000 genes around, and 200,000 RNA. Around 2 million proteins makes one protein pathway. So this kind of, the large amount of the data. So if you are sequencing a one person, the whole genome amounts to be 200 GB, almost equivalent to that, computer, the memory. So the large amount of the data will be involved and the analysis become very, very challenging. That's what the large amount of the data analysis is known as an omics. So the omics technology is important to understand, to practice the personalized radiation therapy. So you are all aware that we are, in general radiation oncology, we are not much familiarized with the genes and genomes because we are almost pertaining to the radiations, though we are medical physicists involved in all kind of the medical practice. But there are common genes involved in radiotherapy response process. So those are just responsible in terms of the context of the first, the genetic variations of RT response. The common genes like EGFR, epidermal growth factor receptor. So that is actually, the function of that cell is the cell proliferation, survival, and transformation. The role of that, the gene is the radiotherapy resistant and the tumor progression. So when the EGFR gene is present in the cancer, probably the resistance will be more, that success of the treatment will be very less. So this kind of, the genes also possible to image not only the genomic, the molecular genomic, but also using the CT, PET, and MR. That is what, quantitative imaging. And similarly, there are many genes like KRAS and ALK in the lung, PTEN the lung and the GBM, IDH in the brain tumor, and PDG-IFRA in the GBM, KI67 lung, breast, and glioma, gastrointestinal stromal disease, and PI3K breast and glioma, MGMT GBM, P53 GBM breast and cancer, and ROS1 lung cancer. So these are all the common genes. It's having different gene functions and have a different role in the response to the radiation therapy. So all this, the process, the genetic or the genomic process can be obtained through the imaging. So what is actually that genomic studies are required to understand or obtain the genomic profile? So the latest the technology is called the next generation sequencing technology will give you that the information about the genomics. So if you look at that, the group of cancer patients comes to the clinic, and if they are subjected to that, the kind of the genomic studies or genomic profiling based on the different the platforms, the genomics that will give you the the genomics and transcriptomics and proteomics and epigenomics and pharmacogenomics. These are all the different kind of the omics technology wherein you will be able to get the the parameter like chromosomal aberrations and amplifications of the the deletion of the DNA, translocations and mutation of the DNA. In the case of the RNA sequencing, you will be able to get that RNA expressions and the protein, the transcripts. So based on this kind of the testing, you will be able to obtain the molecular information. You can classify based on that the molecular subtype of the tumor based on the mutation status or copy number variation, based on that mRNA, the expression, based on the protein expression, based on the DNA methylation, based on the individual genetic makeup. So that entire genome, the characterization or the genetic, the properties or molecular characterizations can be obtained from this, the genomic studies. So I'm not going to go into the details. There are many, the technologies are available in order to get that, the molecular characterizations. If you derive the sample from the tumor tissue and to understand the molecular alterations in the cancer, you can have it, the DNA sequencing and RNA sequencing. In the DNA sequencing, you can get that, the information like the point mutations, copy number variations, or gain, losses or gains, rearrangements and the fusion of the genes and pathogenic sequences, the epigenetic modifications. In case of that RNA sequence, you will be able to obtain that altered transcript expressions level, altered allele specific expressions, and differential alternative splicing, the information. So there are the traditional existing technologies, also emerging technologies like RNA sequencing is very familiar, and the targeted sequencing of the whole exome sequencing, the targeted sequence of whole genome sequencing. These are all that, the very common techniques which will be able to give that, the genomic profiling of the tumor. So this is what actually the technology is all about. So how the precision medicine concept or approach is applied to the radiation oncology. So as I told you, when we are, getting that all the informations, including the clinical and biological informations, the classical, the patient's age and the tumor stage and locations in histology, and the tumor molecular features from the genomic study like mutational status of the tumor and the copy number alteration, gene expression pattern, protein expression, and the tumor imaging feature from that radiomics like locations and the shape and heterogeneity, including the tumor functional profiling from that XYO assays, patients derived model cell lines and xenograph. You will be able to decide about the treatment, whether who is actually required the radiation, who can avoid the radiations. So what is that appropriate radiation dose and field, which agents, in the case of the multidisciplinary the treatment approach, which agents should be combined with the radiation, whether it is chemo first or surgery or the targeted or the immunotherapy. What is the optimal sequence of therapies? So all this, the decision can be taken based on that, the informations of the combined, the genomic and radiomic and the clinical and functional information. Okay, coming to this, what is actually the relevance of the medical physics to this topic to discuss today? So I am not an expert in the the genomic field, but I am also not practicing in the radiomics or radiogenomics. Indeed, we are planning to start, but to understand exactly the veracity of the subject. So I just take this the topic. So how this topic is relevant to that the medical physics challenges today, as per the ISRO. the grand challenges of medical physics are in 4 categories. One is the target definition transformed from art to science. Another one is the development of and the implementation of AI technology in the clinic. Third one is modeling biological effects in the personalized medicine. Fourth one is a visionary leadership. So among these 4, so the target definition and transformed from the art to science is relevant to that. That means that today, whatever the target volume, especially the clinical target volume, or designing the microscopic, the clinical target volume is based on the qualitative nature of that, the imaging. So in case if you are able to use the quantitative imaging, that is nothing but an imaging biomarker, probably that scientific way you will be able to design that CTV, which will give more accurate information to design that, the margin. So this is what one of the main, the concept I emphasize. The report says that, the biomarker based information should be incorporated in the, the design of the CTV. So similarly, modeling the biological effects in the personalized medicine. So what are the, the process, what are the requirements for, the building the model, also important to understand. So this is what actually I have just chosen this topic. So what is motivations, why we need actually in this, the forum? Because, we are all aware that, the medical physics cancer research is not actually, full-time job, because we are also in the service, clinical service. Probably the focusing that, the cancer research is definitely require the dedicated the faculty. That's the one of the, the paper from the Boatfeld and Robert in the PJR, 2011, very critically, the reviewed that, how the clinical medical physics can perform the cancer research. So the recent publications by the Australian, the Physical Engineering, the Medicine Journal, so they emphasize about the medical physics in cancer research. If you look at the different aspects they emphasized, one of the aspects is called the personalization, in that the genomics and radiomics and treatment based on the personal response, adapting the cancer evolution treatment. So these concepts, including the data analytics and the other topics, are discussed, quantitative imaging and molecular imaging. So this is what, the topic of the personalized radiation therapy is important. Similarly, when we are working with that, the clinic, always we give, solutions for the clinical questions. So the recent, the ASTRO, the agenda, research agenda for in radiation oncology in cancer research is, categorized on 6 ways, like the normal tissue and reducing the side effects, the imaging and innovative technologies and expanded indication, the biomarkers and the radiation, and then the tumor microenvironment, combination therapy, big data analysis, patients reported. If you look at that, all this information like imaging and innovative technology, imaging biomarkers, cancer genome. So all this is very much pertaining to that, the precision medicine and personalized radiation therapy. This is that, the need that, the being medical physicists, we are supposed to, involve, understand that personalized radiation therapy, the program. Similarly, in the radiation biology in cancer research, you can see that, the different areas have been assigned by that, the Astro Cancer Radiobiology Task Group. And if you look at that, the tumor microenvironment is one of the important parameter, more than 18% of the component compared to that other parameters. And similarly, the biomarker and radiogenomics also one of the important parameter, whereby you'll be able to do that, the in molecular based characterization for the personalized radiation therapy. So coming to this, what are the resources are required? So are we ready today to practice the personalized radiation therapy? And just I will be giving that, the overview about, what are the requirements. Of course, the biological imaging is possible. In the case of, the PET-CT is very much routine, most of the clinics, wherein that you will be able to assign that, the PET-based, the functional and molecular imaging. And now the MR imaging also is coming up, and probably that will be useful for, designing the biological imaging. And the concept of biological imaging has been introduced way back to 2000 by Ling et al. And the next is that, the quantitative imaging are, the imaging biomarker or genomic biomarkers. That's actually the pertinent to this topic. So one of the approach of the quantitative imaging is, the radiomics. Another one is the radiogenomics. In addition to that quantitative imaging genomic, the biomarker, you are required to have that, the modern practice of radiation oncology, like integrating imaging and therapy systems such as MR-guided linear accelerator and PET-guided linear accelerator is also in emerging. Similarly, in artificial intelligence are almost into the field of radiation oncology, radiology, and medical physics role is in expanding. Similarly, the data science and data sharing and data mining and analytics also is one of the important component which is required for the practicing personalized radiation therapy the program. So the quantitative imaging biomarker is nothing but the biomarker which are objective or quantifiable characteristics of biological process. The goal of imaging biomarker is to quantify the biological characteristics like defining the definitions. In addition to that anatomical and biological imaging, in case if you are able to incorporate that the biomarker information, the dark definitions will be improved. Similarly, that the tumor heterogeneity is one of the limiting factor which is responsible for the treatment failure and the suboptimal treatment. So that is also possible to explore through this the quantitative imaging biomarker. And the tumor microenvironment is one of the important and major factors wherein the lots of informations how that the tumor is interacting with that adjacent microenvironment the tissues, and that is being explored by the quantitative imaging biomarker. So the biomarker properties should be a specific and sensitive enough, and it should be a predictable and stable and non-invasive. The clinical applications of the quantitative imaging biomarker, the diagnosis, staging and patient stratifications, the treatment monitoring, outcome predictions and risk assessment. There are in general two types of the quantitative biomarker, that is a prognostic and predictive biomarker. So these two important biomarkers are important to identify through this quantitative imaging approach such as radiomics and radiogenomics. So if you look at this picture, so this is a kind of the biological quantitative imaging, the biomarker based quantitative imaging, which is able to evaluate the tumor response. Say for example, the tumor microenvironment is possible to evaluate like an MR, the imaging like the diffusion weighted imaging, spectroscopy, and the MT, magnetization transfer, the imaging. Similarly, the cellular density is possible to obtain the information. The proliferation information is possible. The angiogenesis, tumor vessels, the macro and micro structures using the conventional imaging, including that the shape and contour and MR diffusions weighted, elastography, MR anatomical imaging, and MR magnetization transfer, and the texture analysis. And similarly, the specific tumor receptors and protein also is possible to get through that PET imaging and SPECT imaging. The heterogeneity of that the microenvironment is possible to evaluate through that MR and texture analysis and histogram and cluster analysis, and the hypoxia, apoptosis, and water imaging and metabolic reprogramming. So these are the very important component which is responsible to characterize the molecular informations and possible to do that. So this is just the brief overview about that the quantitative MR imaging biomarker. The APM task group 294 report was published in 2021, which states the very long 103 pages of the detailed document to understand about that MR imaging biomarker role in radiation therapy, and put it together. And today's MR imaging biomarker is possible to explore that the biological process like an angiogenesis, perfusion, and hypoxia, the cellular density, proliferation, the tissue microstructure, and metabolism through various the biomarker, which will not be able to much discussed. So another important aspects of the quantitative imaging biomarker, the potential is that in the recent the emerging technology like MR guided radiotherapy, like MR linear accelerator. So this is kind of the MR linear consortium for imaging biomarker working group for the clinical trial. And you can see that the MR linear accelerator, the Unity 1.5 Tesla, that images of the daily ADC map is possible to give you that the molecular and functional imaging, and based on which you will be able to design that the clinical trial. So this process starts like in a biomarker discovery, validate that the prognostic value of that particular biomarker, then that in the technical feasibility through the simulations and planning studies, then the validating that the predictive value through that interventional trials, then the qualification and the selection of the quantitative imaging biomarker for personalized radiotherapy. So this is one of that, the hot area. So I'm not actually, going into the detail, just, give overview about, the radiomics. You all are very much aware, and the radiomics is basically a quantitative imaging approach, and the image analysis method using the computational, the information technology such as artificial intelligence, like machine learning and deep learning. So to define the radiomics, basically the high throughput mining of quantitative image features from the medical imaging that enables the data to be extracted and applied within the clinical decision support system to improve the diagnostic and prognostic and predictive accuracy. So this is what actually the workflow, and in radiomics analysis, the image will be acquired using CT and MRI and PET CT. Once the image has been acquired and the reconstructed images, then the pre-processing will be done. Then after the pre-processing and the image is ready, then volume of interest and the segmentations will be done based on that, the manual method as well as semi-automated and fully automated. And once that volume of interest has been defined, then the volume of interest will be given to that, the feature extracting, software algorithm, which will be working based on that, the four important, the parameters like geometric shape and, the histogram of, the statistical histogram and texture based and filter based. And once it has been, made that, the computed that the features, then the feature selection should be done. For the feature selections, there are different approaches are available. The statistical threshold approach is called, the ICC and intercorrelation coefficient and coefficient variation. Another is data driven, like the principal component analysis, and at least the shrinkage and the selections objective, the operator algorithm. So this is a very important algorithm for utilizing in the feature selection. And once the feature has been selected and qualification is made, then this informations will be given to that, the classifier in the model, for the modeling purpose. The classifier will be dealt by the machine learning, the algorithm such as support vector machines, the logistic regressions, and the random forest, etc. So using this, the machine learning, one can classify about the risk assessment of the patients. So this is for, the hand grafted, the based the radiomics. And once if you use this informations directly to the deep learning, deep learning module, it will be like a black box. You just give an input, and all the informations and the process will take place based on that, the deep learning, and mainly that convolution neural network deep learning is used for extracting the information and modeling. So this is kind of the overview about how the radiomics actually works. And once you derive that the genomic parameters from that the radiomics, that's called the radiogenomics. So the main important the aspects of radiomics is how to extract that in the radiomics feature. So what are the methods are available? So this is one of the important the features like geometric or shape based feature, the first order the feature statistical histogram like mean value and median value and 10% and 90% percentile difference of the intensity of the particular pixel or voxel, and image texture whether it is a coarse or fine, and image filter based on that the edge detection and the edge emphasis and length scale and low gray emphasis. So these are all the different the methods one can derive that the radiomic feature. So there are multiple, there are more than the 100 and to around 100 features are available in the literatures, which I simply put it in that way of understanding, and it will not be discussed much. But you can see that this category can be classified in two ways. One is that static feature. Another one is that dynamic feature. So the dynamic feature example is that the blood flow and regional blood volume, the mean transit time and extraction fraction, and permeability or surface area product, volume transfer constant, extravascular, extracellular volume. So these are all the dynamic, the radiomic features needs to be obtained. it's a bit complex compared to that static feature. In the static feature, we can classify, the morphology based or intensity based. In morphology based, example like, the volume based and surface and, the area and sphericity. Similarly, intensity based can be classified like a first order, second order, higher order. In the first order, the example like a mean percentile and standard deviation and kurtosis and, the skewness. In the intensity based second order is an important feature like a gray level co-occurrence matrix based feature, gray level run length matrix based feature, gray level size zone matrix based feature, gray level distance zone matrix feature, neighborhood gray tone difference zone matrix based feature. And the examples are like, auto correlation, the cluster prominence, difference variation, homogeneity. Similarly, that example for the gray level is in a short level emphasize, run length, and these are all the examples and the features one has to really compute that features to understand what is the feature involved. So coming to this, what are the factors influencing the radiation mix stability? So I'm not going to go into the detail. You can see that, the different parameters in the image acquisition, in the image reconstruction, segmentation, post-processing and feature extractions are possible, or it is required to, it will be influencing that, the radiomic stability. So ultimately, that, radiomic feature should be a robust enough and it should be a reproducible, and that, the performance in case of the model performance and classification performance should be accurate enough. So there are various parameters in MRI and CT and PET. So these parameters are important, which will influence in the radiomics feature stability. So there are a lot of resources for radiomic studies. The open source toolkits are available, and many people are aware. One of the very common tool is called the Radiomics, and the extension of the Radiomics, another one is called the Pyrex. And Radiomics and Pyrex are using that Python 3D slicer as well as the declare-based platform. And imaging biomarker explorer or IBEX, another important the open source toolkit, and Windows or MATLAB based. And Stanford University have developed a quantitative imaging feature engine algorithm based on a MATLAB, and the Radiomics license, cancer imaging phenomics, and the deep neuro and the PET oncology radiomics. There are many, the source in-house also is available for performing this, the radiomic features. So similarly, there are the open data sets like Radiomics Digital Phantom is available. The University of Texas, the Credence, the cartridge radiomic phantoms are available, and quantitative translational imaging medicine phantom and data set, and the cancer imaging archive phantoms are available. So all these informations are possible to obtain in open sources. So it is very much important to mention and share that one of the criteria for the radiomic features to be qualified for the clinical utility is the standardization. To ensure the standardization, the International Standardization Biomarker Initiative has been taken by the group of physicists, and they have given that certain criteria and how that the feature, the radiomic feature needs to be extracted. That that has been ranked through that radiomics quality score (RQS), and in this you will be able to meet out the criteria. Then probably you will be able to use that biomarker in the clinical trial. So similarly, that the NCIUN, the Quantitative Imaging Network, and similarly RSNA, the Quantitative Imaging Biomarker Alliance, is important resources whereby we will be able to obtain that image information regarding the radiomics and radiomic quantitative biomarkers. So very important to the consortium is responsible, or the working on the radiogenomics project. One is called the Radiogenomics Consortium, which was started in the UK in 2009. The later it was actually established by the NCA US in 2009. And then another one, the European, the project called the Required. So another one important, radiogenomic consortium is possible. It's the ongoing. So there are a lot of information, clinical information is possible to do that. So these are the radiomic phantoms, which I told you. So what is the current status of the radiomic literatures? You can see that the growing radiomic studies until 2016, only 40 articles were there. In that the year 2021, So the 1,145 the articles are being published in the radiomics the field. So if you look at that in the area of the cancer type, the breast is being the topmost area wherein that the radiomics studies are carrying out. Then that the genitourinary prostate kind of the gynecological head and neck and neurological and etc. So this is actually published in 2013. That's what the half of the year it was not included. The 2022 is only showing the less. It doesn't mean that it is actually decreasing. Okay, coming to this, what is that the status of AI radiogenomic studies? If you look at that, one of the recent the publications in cancer. So the author has mentioned that the radiogenomics publication over a year is not much. Up to the year 2021, the number of publication is up to 15 maximum in the 2018, and the country-wise distribution of the radiogenomic studies, the US is maximum 39%, the contribution. Next to the China is 31%. Then all other Japan and Germany and all other countries are the contribution of the radiogenomic the research studies. Similarly, if you look at that the machine learning algorithm is being most commonly used, and next is the deep learning, the 33%. Then the various the machine learning algorithm is being used, including the CNN, and different models are being used. And if you look at that, what are the imaging modalities? MRI is being used to 45% of the imaging modalities for the radiogenomics, and CT was 39%, and then the CT and PET is 8%. Similarly, if you look at that, the radiogenomic, the studies in clinical, the brain is maximum, the 23%, then the breast is 14, and lung is 15, and liver is, the 8%, and remaining this one. So coming to this, the radiomic-guided radiation therapy opportunities, I'm not actually, I have not done any work, and also this, the review paper given that, kind of the stimulations to share with the people of our own community that, how the radiomic potential is going to be in the future. If you look at that, radiomics-guided radiotherapy is possible during, pre-treatment, for the treatment selection, patient selection, and treatment planning, and during the treatment or intra-treatment, treatment delivery, treatment verification, adaptive radiotherapy, post-treatment treatment, the toxicity assessment and treatment assessment and response differentiation. So this is what the concept of radiomic based target volume has been introduced. And if you look at that, if you obtain that imaging from the MR and PET, once you delineate that, the physician delineate the target volume, this volume will be given to that radiomic feature software. So that will extract from that, kind of the tumor plus surrounding tissues based on the texture value, based on that voxel intensity distribution and wavelet, and also from the contours of the tumor using advanced tumor shape. So if you look at that in the box, this represent that, the tumor, and this is the contour. So you can extract that in the different heterogeneous informations based on this radiomic feature. So you can see that the red is basically the high risk, whereas the green is a low risk. So this is what you can boost the dose in the higher area, that is called the radiomic target volume. In the green area, it is the less heterogeneous. You can give less dose, or you can give that conventional dose. So this is the concept of radiomic, the target volume. So one of the hypothesis authors of this, the article published in the Physics of Medicine and Biology in 2022 in December, they says that this concept of radiomic-guided radiotherapy is possible to identify cancer hallmarks that involve with the radiation therapy response, like imaging phenotypes directly reflecting the underlying biological response or genomics. You can see that the texture analysis of the intensity GLCM, GRLCM, and all this, the texture, the imaging, the radiomic feature is directly possible to correlate with that, the tumor or the cancer hallmark, like an evading growth suppressions and the cancer hallmark. Okay, so this is one of the important, the publications one can refer, and this is a very detailed review article. And similarly, you can see that when you obtain that, radiomic target volume, you can also obtain that, the sub volumes. There are 11, the different regions. You can, based on the radiomic feature, you can categorize the different region of the, sub volume of radiomics. Then that it means that, the heterogeneities are different from the different within that, the radiomics volume. You can, optimally give the dose to the higher, the heterogeneous area, and less dose to the, the less heterogeneous area. And this is how one can also incorporate this, the radiomic, the signature map in the optimization. Say, for example, knowledge-based planning is possible. This radiomics knowledge can be incorporated into the TPS. You can give that, the radiomic volumetric signature map, similar to the intensity influence map. You will be able to, design that in each and every angle, and you can obtain that, the radiomics knowledge-based planning. And similarly, you are able to extract the information from the radiomics, predicted TCP, NTCP. You can, incorporate that information to obtain that, the tumor control probability based on the radiomic features. So again, the dose painting by feature is possible, similar to the dose painting by contour and dose painting by, the points. So dose painting by feature is also possible based on that, the GLSM, the feature layer, the regions. You will be able to, give that, the painting to that, molecular area. So what are the challenges of, the radiogenomics? So the radiogenomics basically the suffers based on the, because of that, the standardizations and harmonization. Because radiomics and radiogenomics workflow in the clinical routine is still as challenging. It's not actually in the routine. So this is one of the reason, why this the radiomics and radiogenomics area is still in the research mode. Because the reproducibility of the extracting the radiomics feature is actually challenging. Then the model overfitting, because if you are giving less number of, the training and the, the validating data, probably that the model will overfit and it will predict the accuracy less. So the prediction accuracy is very much important and challenging, and it is required the multidisciplinary. One person cannot, involve in this discipline. The clinician needs to be there to target, and the data scientist needs to be there, imaging scientist needs to be there. So the imaging physicist needs to, all these people need to be there together and need to collect the large data collection. So the data is very much important. Small amount of data is not possible to rely upon any kind of an accuracy. Similarly, the integration of the radiomics modeling into overall, end-to-end QA is becoming very challenging. And the radiomic data and the algorithm validations into the TPS also is challenging, and quality control of all the technologies is one of the very challenging. So what is that new opportunities based on this, the radiomic and radiogenomics? There is a possibility of now the prevention of that disease, because the less patients can be treated. Because of this, the molecular characterizations, you will be able to classify, the favorable group of patients, unfavorable group of cohort patients. So probably the potential for improved access to the resources. Similarly, so early detection is possible when once you are able to attain or attempt to the molecular informations, and more early tumors can be treated. It can be of competitions with like the surgery and endoscopy in the case of the esophageal and GI cancer, and it is a minimally invasive. And also the improved treatment is possible because the multidisciplinary approaches, the high precision automation and digitalization and integration of the biology is possible. These are the new opportunities. So coming to this, what is the feature of medical physics in the precision medicine? I just put this slide based on my experience of this the review, and in the process of formulating kind of the subspecialties of medical physics. You can see that the different area of additional new is important to cover up, to understand, to work as a medical physicist in the precision medicine or personalized radiation therapy. Look at that, the radiation dosimetry as we do it, and radiation technology and medical imaging and radiation therapy. Apart from that, not only that, the radiation biology, cancer biology is important, cancer genomics is important, and cancer immunology, integrative mathematical oncology, computing, artificial intelligence, and data science. These 10 important fields are required to be the medical physicist, in the precision medicine or personalized radiation therapy. In fact, this is what, I am in the proposal of, proposing to establish the super specialty of, the medical physics discipline called the oncology medical physics in our, country. And why do we need, that the need for the translational research, is, very much important. 100% diagnosed with the cancer, 70% is, the local, regional, and 30% metastatic. Out of 70%, 30% can be treated with radiation, 35 without radiation. Out of 35%, 25% can be achieved the local control, 10% can be the failed, it's a failed due to the local recurrence, and then, this one. So if you look at that 5% of failure, physical cause 5%, if you look at that, how much the proportion of that 5% will be huge amount. That's what, the biological and, the physics is important. Coming to this summary, the radiomics is a quantitative imaging biomarker. The radiogenomics is a combination of both imaging and genetic biomarkers. The radiogenomics is a translational radiobiology research field towards the precision medicine in oncology. It will improve the diagnostic and predictive power in support of the clinical decision making for personalized cancer care. The biomarker driven, the translational research, an important contribution in cancer care towards the precision medicine. So the translational research is a traditional role of medical physics in the medical discipline. Thank you very much for your patience listening.
Thank you, Dr. Subramani, for an excellent overview of radiogenomics into personalized radiotherapy. The current status, you have tried to elaborate the challenges and the future opportunities. Huge opportunities are there, and this is the area shows the amount of publication and these things. And definitely, this will improve the outcome of the radiotherapy. And we very well know that it is not the size of the tumor, but it's the biology of the tumor which matters into the final prognosis, and how this radiomics and genomics can be used for diagnosis, treatment, and even the prognosis of the disease and all these things. I think you can stop sharing now. And there are a lot of questions. Before I take up the question, lot of people have asked, this webinar is CPD accredited, and depending upon your attendance, you will get the certificate. Second thing, the recording of this webinar will be on the IOMP website. As usual, all the earlier webinars are there on the IOMP website, and this will be also there. Now, I will put few questions. First question is, does radiogenomics can be combined with immunotherapy? Because your last slide, you talked about the immunology and other things. Can it be combined with immunotherapy?
Yes, there are models. The immunological model, or the immunoresponse model, is possible. And again, the complexity of the model building and model validations and model accuracy becomes a challenging. But it is possible to incorporate the radiomics based the information into the immunotherapy practice or immunotherapy response evaluation.
Okay. Another question is, are some tracers used in radiogenomics to track the genes? If yes, then what are they? Are there any, I don't think any tracers to track genes. What is your say?
What is that? The tracer? No, it is basically based on that, the imaging features. Yeah. The imaging features are nothing but the phenotypes. That phenotypes needs to correlate into the genotypes. So that is actually the tracer, what I mean to say. So that's kind of the correlation from the imaging features.
Okay. There's another question. There is more area in omics. It is a metabolomics. How cancer metabolism affects cancer treatment response?
Yes. So the metabolism or metabolic, the profiling is important to understand, the biochemical process of the particular, the biology of the tumor. If you are in a position to understand the characterization of the biochemical process through that metabolites, and to target that metabolites, the amino acids, and the glucose. So these information, the targeting the cancer metabolism is one of the important, the objective for the personalized radiation therapy. So the metabolites or metabolomics information is very much important to incorporate into the, the personalized radiation therapy.
There's another question. Are there any commercial TPS, treatment planning system, available for taking into account this radiomics and radiogenomics for treatment planning?
To my knowledge, there is no commercial treatment planning system. However, there is a group from US, Srikar et al. They have published, the article in 2016 that's called Radiomics-Targeted treatment planning. They have designed that, the module 3 module, which will be describing that, the brachytherapy of external beam and, the brachytherapy in external beam. And each module will be possible to calculate, the treatment planning based on the radiomics data. So that's called the RAD trap, the framework. That's what my knowledge. Apart from that, I do not have any knowledge on that, the commercial availability of the treatment planning system as of today.
Okay, thank you. There is another question, multiple times has been asked. How does genomics sequencing affect the time frame prior to radiotherapy? How much time does it take before we start real radiotherapy planning, and the time lost between the diagnosis and the treatment, and is an intervention during and or after the treatment? This is an important question has been put.
Yeah, the timing for the genomics, initially more than 11 weeks were there actually in the year 2007. If you can see today, it will be in hours time that the genomic profile is possible to analyze, number one. Number two, the conventional, the genomic, profile is little bit of, the time consuming in order to incorporate those informations into the routine clinical workflow. So that is one of the reasons that, the imaging quantitative biomarkers, such as, the MR linear accelerator and PET-LINAR, is possible to extract the information during the treatment or prior to the treatment. So that can be correlated, and that is possible to do it. In the conventional, the method of genomic profiling, probably it's not, challenging to, incorporate into the routine clinical. That's what my view is.
Okay, there is another question. Radiogenomics and radiobiology, there are any new radioprotector drugs or radiation protection point of view? Somebody has asked about.
I do not have any much knowledge on the drugs. Probably, we can visit to that, what kind of the drug will be available in the later.
I think we are exceeding time. There are questions and things, and because we stick to our time limits and these things. So once again, I thank all the delegates. Almost the number touched to 780, very good number, and a lot of good comments they have come up. Large data, large knowledge, and slides into very short span of time. So thanks to the speaker, Dr. Subramani. I thank once again to Professor John Damilicus, IOMP President, for giving me the opportunity. And Magdalena has strongly background support and this thing, which made this webinar successful as usual. Everything, anything you ask, immediately she will be available. Thanks to Magdalena, every team of my IOMP, all chairs and this thing, thanks to them. Thanks to you, Dr. Subramani, for sparing time and voluminous data giving it. And I think now the delegates and who have missed this also can watch it repeatedly on the IOMP website and get insight into this very exciting, very challenging, very useful the technology, and it is actually convergence of many sciences, many technologies for betterment of a personalized radiotherapy. Thank you, one and all. Thank you, Dr. Subramani, for excellent talk and sparing the time. Thank you.
Thank you, Arun, for organizing this webinar. Thank you, Dr. Subramani, for your excellent talk. Thank everyone, the audience, for their presence and active participation. Thanks a lot.